So, Charushree is an MTech graduate in clinical engineering from IIT Madras with research interests in microplastics and their impacts on human health. She is passionate about interdisciplinary research at the intersection of environmental toxicology and healthcare. And today, Charushree is going to talk to us about detecting microplastics in biosamples, a bridge between the environmental toxicology and clinical science. So, thank you so much, Charushree, for being here. And I will just hand over to you.
Hi everyone. I would like to start with a thank note for ASN25 organizers for giving me this opportunity. And here is my topic that is about the microplastics, the major growing concern. My work focuses on detecting the microplastics in the human biomarkers, and it's quite deviated from the actual theme of Anthropocene sediments, but that also seeks to understand how the human made sediments are sourced, transported, distributed, and redistributed, as well as their final destination. And in the essence, I was introducing the microplastics, which is 1 micrometer to 5 millimeter in size, and the nanoplastics is the microplastic size, which is less than 1 micrometer, can be classified as the nanoplastics. And they are forming from the breakdown of the larger plastics, shredding, leaching, that is called the secondary, or directly from the manufacturing of the microplastic itself for their need. It's called the primary sources of the microplastics. And their sources include the consumer products like synthetic clothes and plastics packaging, and more the industrial processes and healthcare laboratory are the major source of secreting the microplastics. The healthcare industry is being the major source in contributing the environmental microplastics than any other things because of the trade-off between the contamination, the sterilization, and single use of the plastic particles. In essence, Anthropocene microplastics signifies the pervasive presence of tiny plastic particles as a defining characteristics of our current human-dominated geological era. They don't stay put, they transported globally through various medias. Microplastics are not any some distant pollutant. They were being intertwined on our daily living things. They are in air we breathe, water we drink, and the food chain that sustains us. The persistence of the plastics in environment is now being a major concern, contributing to the pollution, harming the wildlife, and potentially increasing our impacting our ecosystem. And here is the key aspect: systemic dimensions and long term consideration of the Anthropocene microplastics. The key aspects includes the pervasiveness, source diversity, longevity, ecological impact, and lifestyle connections, which means the pervasiveness that is present everywhere. It is inevitable and ubiquitous. Source diversity, in the sense, it is produced from various sources from our day to day life, and the longevity is its persistence and accumulation over time to create the long term concerns. Ecological impacts as it affects the ecosystems, human health, and the rising concerns. And the lifestyle connection in our day to day and individual responsibility mm, made the made this. Systemic dimensions include the global transport, which they were traveling through air, water, to even the remote ecosystems, and they were entering into our food web, impacting the lower organism to the tertiary organisms in the food chain, like the humans, and they are being the pollutant vector to concentrate and transport the other pollutants, such as the additives, and rooted for unsustainable production and consumption patterns. Ethical implications include our responsibility towards the future generations, safety of the future generation, to reduce the use of the plastics. And the long-term indications includes the geological marker as it signifies the human-dominated epoch after the 20th century. The plastic production have been increased, and the ecosystem disruption impacts across the different trophic levels, and various exposure pathways leads to potential health concerns. And the systemic unsustainability is the critic for linear economic models, and exploration of the circular economy is important. And ethical imperatives include environmental justice and intergenerational equity for our future generation. So generally in academic terms, The Anthropocene microplastics demand a critical and interdisciplinary approach that involves not only quantifying and the characteristics of the pollution, but also analyzing the systemic causes, long term consequences, and the ethical implications on it. Microplastics are being the emerging pollutants, and they were being increasingly recognized as the key stratigraphic marker for the Anthropocene approach, with their invasion into their human environment and tightly linked into the sedimentological network. Addressing the MP's pollution requires the systemic changes, moving towards the circular economy model, reducing plastic consumption, and improving the waste management infrastructure globally. This image shown here depicts the pervasiveness and influence of the trophic level in the pollution. The following slide focuses on the exposure of MPs on human biological system. This is showing the exposure and effects in our biological system. Biomagnification and bioaccumulation are the two major pathways of concern when it comes to human exposure to the MP pollution. Biomagnification is the term where MPs can be associated with the toxins increasing in concentration as they move up towards in the food chain, uh, reaching humans from the lower level organisms, since humans were the tertiary consumers of every food product. And bioaccumulation means the gradual buildups over the time. This neurotoxicity, nephrotoxicity, hepatotoxicity, and hemotoxicity were the toxicity of the pollutants caused inside our internal organs. We are living in the era of artificial organs where our natural organs are being continually deteriorating by such toxic pollutants. Toxicokinetics means absorption, disruption, metabolism, and excretion of the pollutants, whatever we are inhaling or ingesting or accumulating through infusion or transcutaneous invasion. And everything is being cleared by our excretory organs. The beyond the clearance, the burden persists due to the extreme pollutants. This is what we want to quantify. What are the bioburden we are having? And this is one of the results obtained on analysis of the bottled drinking water that I uh, that shares the leachate of various polymers that were not even visible to our naked eyes. But these kind of pollutants, which we in the drinking water, are the major concern over our day-to-day life, which is an alarming situation. And these kind of studies leads to various another studies focusing on MP's reduction in the human samples. And these are the key findings of the micro nanoplastics detection in human biological samples. The term microplastic itself coined by the marine biologist named Richard Thompson from the University of Plymouth in 2004. After that, many research have been underwent and concluded that MNP's contamination in environmental samples such as river sediments, beach sands, road dust, etc. had been found. And even in the internal organs like intestines, lungs of the marine organisms like fish, lobster, zebrafish, phytoplankton, zooplankton, and so on. And the research had been continued on the samples since 2018 in the human samples. The first detection of MNPs in human stool samples was conducted in 2018. After that, 6 major microplastics from polyethylene terephthalate, polystyrene, ethylene, polyvinyl carbonate, and polyurethane have been found in the human blood out of 17 to 22 healthy volunteers and quantified that 1.16 microgram of microplastic particles present there in the 1 ml of blood. This is the groundbreaking research study which states the circulating peripheral venous blood is containing the toxic pollutants. And in 2024, the review literature stated 8 out of 12 major organ systems in human have detected the presence of microplastics. These have been detected including blood, liver, heart, kidney, brains, lungs, spleen, reproductive organs include placenta, amniotic fluid, semen, and in body fluids. And this is the importance for knowing the biomarker detection in the microplastics. Biomarkers include blood, urine, hairs and nails, and other internal organs, the biological fluids, and anatomical waste. So far, beyond the study, almost every organ from our organ system is found to have that exposure. And there are also some drawbacks, which includes when we detect MNPs in the excretory organisms or anatomical waste, it also shows that we were again giving the microplastics into the source from where we get into it. We need to, there is a need for getting the long-term concern of the microplastics in the human exposure. And this kind of comparative studies between the different biomarkers and knowing the necessity is choosing appropriate biomarker for human exposure studies is being important. Studies that concluded the presence of MNPs in amniotic fluid, semen, placenta, and meconium had proven that we are exposed to such pollutants even from our fetal life. The biological sediments, such as plastics in human tissue, has been seen as the living stratigraphy of Anthropocene. And this is the scientific and social relevance of why we need that study. This contribution leads to establish a dose-response relationship for the micro nanoplastics exposure akin to heavy metals and organopollutants. These things like heavy metal and organopollutants have been standardized method and been established beyond the decades. But this microplastic study is still been in infancy, and we need to uh, put up a standardized method and analytical methods for quantifying the presence in our system. And for regulatory and industry standards, informing about the exposure limits on workers and vulnerable population like fetals is very important because they can develop a developmental neurology effects from their birth itself. So limits include ALARA principle, which is ambiguous to radiation safety, like time distance shield optimization. These kind of studies were well established, but these things also need to be established in case of toxins such as MNPs. This core philosophy is to minimize the harm through practical and theoretical actions of reducing the plastic pollutions. And occupational safety and environmental safety has to be developed, and the policy development for the regulatory bodies defining the exposure limits, monitoring framework, and interventions have to be designed for such the major crisis of toxins. And this is the end of my presentation. As a clinical engineering, I view the Anthropocene not only through the lens of the environmental change, but through the emerging biomedical dimensions. MPs are no longer confined to oceans and soils. They are now detected in human tissues also. In that sense, we are becoming the part of the biological sediment record of our time over the time. I'm open to domain this research and explore the intersection between the environmental toxicology and clinical science. Thanking you.